So I mentioned last time that there are a few traits that in humans are considered to be dominant recessive. This is a list of some of them. And the thing about genetics is that it's never, or almost never, as simple as it seems, that this does not necessarily give you absolute predictive ability over combinations of human traits. And most traits are not a simple, yeah, the protein works, the protein doesn't work, kind of dominant recessive pairing. You get a lot of proteins that are maybe a little bit different, that have slightly different effects, and the effects kind of mix together in various ways. You have a tremendous number of features where you can't eliminate it down to just one protein working, that it's a product of multiple proteins, so multiple genes are at play. But we're going to continue down on the history of genetics. So people started to build on Mendel's work. And one of the things that Mendel discovered in his pea plants was that no matter how many of the different traits he fiddled with, they never seemed to affect each other, is that they always were inherited separate. He called it the Law of Independent Assortment. As people started to build, they found that that was true for some genes in some organisms. They weren't always looking at pea plants. But it wasn't always true, that some genes were passed on as combinations, as they were connected to one another, even if they weren't for the same trait, and it wasn't obvious in any real way why the traits would be connected. But they were somehow linked. And that's the next discovery, going into maybe the 1920s. Now what I'm going to do with this is I'm going to talk about the discovery, and I'm also going to talk about what it actually means that we know now. So I'm going to talk about chromosomes. Took a long time to link linkage to this. In reproduction, you pass codes on to your offspring. You may pass copies in asexual reproduction. You may mix them with one set of copies from another source for sexual reproduction. But there's lots and lots and lots and lots of genes. And for any type of reproduction, including just making more cells, you've got to make sure that you make copies of the codes, and that one set of copies goes to one offspring or one new cell, one set goes to the other new cell. If all of our codes were separate pieces of DNA, and say nobody can really decide how many genes a human being has, but somewhere between 12 and 15,000 is the general number that you see. So say we got 12,000 genes, we got a maternal one and a paternal one of each one, that gives you 24,000 to 30,000 genes. If they were all separate pieces of DNA, to make those copies and distribute them properly would be a nightmare. You would mess it up a tremendous amount of the time. What linking genes together does in a chromosome gives you a manageable number of DNA pieces to make copies of and move around when you need to distribute them to new cells or to offspring. Species, one of the things that can define a species is that each species has its own particular chromosome number. Humans have 46 chromosomes, 23 pairs, 23 paternal, 23 maternal, combined into 46. That's the chromosome number for humans. Chimpanzees have 48. You can actually, because you go, oh, but we had the same ancestors. Somewhere between the ancestor that went to chimps and went to us, a pair of chromosomes fused at the ends on our family tree and reduced the 48 number down to the 46, even though we still got those same genes. Instead of being on 2 separate chromosomes, they're on 1 chromosome for humans. And you can tell that by homology between the genes and what the genes do. Anyway, chromosome numbers can range from a fairly low number to a fairly high number. In sexually reproducing species, it'll be an even number because you send sets on. In prokaryotes, it's simple. Chromosome number is 1. Bacteria have a single chromosome. And we'll talk about that later in the semester. But we have 46, 23 pairs. Some animals have as few as 4, just 2 pairs. Some plants have hundreds. Plants are a little bit more easygoing in terms of not getting the copies to go where they're supposed to, so they can kind of get by with a higher chromosome number. You'll see what that means in a minute. A roundworm with only 4 chromosomes as its chromosome number. When they're dividing cells, when they're making sperm cells, when they're making egg cells, when they're making body cells, moving 4 chromosomes around, you're not going to mess that up very often. So you don't make as many mistakes. And a mistake of that is a type of mutation. But there's only so many ways you can combine that low a number of pieces of DNA. So you don't get as much variation in your offspring. There's not that many ways to mix afterward. Now, I'm not going to write the rest of this down, but you can figure this out. This often happens where the advantage of one approach, when you got 2 ways to do something, the advantage of one reflects on the disadvantage of the other one, like sexual and asexual reproduction. So with high chromosome numbers, you're going to make more mistakes. But you're going to wind up with way more ways to mix those numbers together. So we've got 46 chromosomes. That's a relatively high number for animals. And Down syndrome is a mistake where chromosomes don't get distributed properly. As women get older, they have more trouble getting pregnant. That actually isn't true, but if you wind up with chromosome errors in a sperm and egg combination, most of those chromosome errors, the embryo will die very quickly. And you probably won't even know that there was ever a technical conception. In fact, an extra number 21 chromosome, which is Down syndrome, and some stuff with extra X's and Y's are pretty much the only survivable extra chromosome events in human conception. So we make more, and those egg cells are getting older and older and older. They're going to make more mistakes, so that's why it's harder for, Down syndrome is more common in older women, and apparent issues with conception are higher in older women. But you're just going to make more mistakes, but you get more variation, right? Variation's a great thing for producing offspring with variation, that's how evolution is driven. So that's a really good thing. Now these are not things that they discovered at this point in time. I'm just kind of building on linkage connected to chromosome, what chromosomes mean and what the numbers mean, because it's just kind of easy to interject them here. So they revised Mendel's laws and went, sometimes these genes are going together as packages. Sometimes these alleles are connected to one another. That's how they knew. Different alleles were traveling as packages onto offspring, showing that those stretches of DNA were somehow connected. But the more they did research, the more they found even that couldn't be trusted all the time. That sometimes the connection was broken, and alleles that had been connected and connected and connected suddenly weren't again anymore. And it, again, took a really long time to find out that during the production of sperm and eggs, that when chromosomes get really close to one another, they sometimes swap pieces. So if there's two connected alleles on this chromosome that don't exist on that chromosome, and this piece gets traded over there, this piece gets traded over there, then this allele is no longer linked to this one because it's on the other chromosome. So if this one goes on to the offspring, the linkage is broken. If this one goes on to the offspring, the linkage is broken. The other linkage is broken. This is all stuff that gets covered later when we actually get into cell division and meiosis, which is this type of cell division. So it's kind of introductory. If I went fast and that was confusing, the idea that it breaks linkage is kind of what you need to know at this point. In the United States, people discovered that Drosophila Melanogaster, very easy to keep in the lab type of fruit fly, that they sometimes you'd look at like you'd have fruit flies. I don't know why people were raising fruit flies before they realized this, but you'd see features that would just kind of pop out of nowhere. Like their eyes would change color, or their markings would change color, or they'd wind up with malformed wings, or all sorts of different things. And to one extent or another, those changes were inheritable. Very often they were recessive inheritable because they represented very often a mess up in what those coded proteins were doing. But it was discovered pretty quickly that in these little animals that you could keep a lot of in the lab, so you could have high numbers and they were easy to deal with, and you could see them through a dissecting scope really easily, that they were a good model organism for the basics of genetics. And what was making changes that you didn't see in the population? What was making the little bitty wings, or change in body shape, or change in eye color? They were mutating. That's the next thing on the historical list. Now again, we look at mutations and go, oh, it's a change in the DNA. They did not know that. They did not know that DNA was connected to genetics. People suspected, but here was the problem with DNA. Its code is in sequence, but nobody had any way of analyzing sequences. And if you break DNA down, it seems like a very simple molecule. It's got just a few components, and nobody could come up with any possible chemical way that could store huge gobs of information. So when they said mutations, they eventually started to talk about that being changes in DNA. But here it was just changes in genes when they didn't really know what the genes were. But they discovered some stuff. They found out once mutations would appear spontaneously in fruit flies, obviously there are people going, well, let's see if we can make that happen. And they found that exposing fruit flies to certain chemicals would increase the amount of mutations you would get. By zapping them with certain types of ionizing radiation, it would increase the types of, it would increase the number of mutations that you would get. And so then in other areas where people were looking at other aspects of genetics, this idea that this chemical might get you more mutations, or that type of radiation might get you more mutations, had, again, wider impact. People read the papers and went, oh, let's do that with our model organisms. So this was fruit flies. I said Drosophila Melanogaster is that people who work on these things, or even talk about them, almost never call them Drosophila Melanogaster, even though there's 30-some different species of Drosophila. When people say Drosophila, they kind of, everybody knows which fruit fly you're talking about, unless you're working for the agriculture department and you're actually dealing with a different species of fruit fly that's a pest. And even though Drosophila is a genus, It very often is not written as italics or underlined, where Neurospora, which is the genus of a particular type of mold, usually is italicized when you see it written. For this semester, we're trying to keep the chemistry at a minimum, but there's some stuff you can't avoid. Enzymes. Enzymes are a type of protein that basically are necessary to get any kind of chemical reaction working in cells. They're kind of the chemistry boosters of living things. And people were working with this mold, Neurospora or Neurospora, and they had worked out a lot of the chemical step-by-step processes, the metabolic pathways in these particular molds. And for each one of the pathways, for a bunch of the pathways at least, they had figured out, okay, this is the enzyme working on to get step 1 to happen, this is the enzyme working to get step 2 to happen, this is the enzyme working to step 3 to happen, this is the enzyme working to step 4 to happen. Now keep in mind, nobody knew how the mutations were changing what they were seeing, but what the Neurospora people had was they had a broader foundation of knowledge. And when they caused mutations in Neurospora, they could figure out, okay, this feature is different. This pathway is messed up. Where is the pathway messed up? Enzyme number 1, still working okay. Enzyme number 2, still working okay. Enzyme number 3, ooh, we're not getting the product from enzyme number 3. Enzyme number 3 must be what's messing up this whole pathway. It's not working right because we mutated it. Enzymes are proteins. These mutations they were seeing were specifically changing single proteins in the pathways. So this got us closer to figuring out just exactly what genes were. We didn't know they were DNA yet, but now people started to look for other mutations that could be connected to non-enzyme proteins and found that's what was going on. When you saw a change in a feature, if you knew enough about how that feature was produced, you could almost always link the change to a single change in a single protein. And people knew how important enzymes were to everything that happens in a cell. So, but there are other proteins working as well. And so they figured out different changes in different proteins, that's what the mutations are doing. We'll get one more thing on this list. People were studying a type of virus that invades bacterial cells. As they were figuring out bacterial cells, the chemistry is a little bit simpler, so it's a little bit easier to figure out what's messing with it. And they were finding out how viruses work when they invade bacterial cells and take them over to make them into virus producers. Because we've talked about this already. Viruses get their packaging into a cell and turn the cell into a virus factory. What they discovered for phages is that the only thing that's getting from the phage into the host cell is DNA. Now we now know that some viruses are more complex than that some of them also bring in some of their own enzymes, because there's a couple of things that they do that isn't just using the host's DNA processing machinery. But everything starts simple. But this was a really big discovery. What do you need to make another virus? Well, it turns out all you need is DNA. So when people were trying to figure out what's coding for the proteins that make the virus and actually are parts of the virus, so they could go, well, it's DNA. And that's where we'll stop this one. We'll pick up Watson and Crick next time.